In this second video of the endomembrane system, I will be discussing the source of the production of a majority of the products from inside the cell, and that's the endoplasmic reticulum. So the endoplasmic reticulum, as we know, that is actually subdivided into two parts based on their function and also on their appearance. So first we have the rough endoplasmic reticulum, which appear rough because of the presence of ribosomes on their outer membrane surface, and the smooth endoplasmic reticulum because they lack ribosomes. What's more is that the smooth endoplasmic reticulum are tubular in shape, as we see here, while the rough endoplasmic reticulum are flattened in shape. Now, here's a picture of the rough endoplasmic reticulum, and here's a picture of a smooth endoplasmic reticulum. So the name itself means endo, meaning inside the plasma membrane or in the cytoplasm, endoplasmic, and reticula meaning reticulated, sort of like wavy, curved, in like a snake, okay, so reticulated, and heavily folded, okay, so that's where the name endoplasmic reticulum would actually come in. So here we have a cell. This one could be a secretory cell that would be producing a hormone, a protein hormone. So the ER are actually the ones responsible for the production of the protein hormone. And the cell here is outlined in blue, and we can see that it's actually polar, meaning to say that there's an end of the cell that is facing the duct, okay, where the final product, the hormone, is released. And then there's a part of the cell that's facing away from the duct, perhaps attached to a basement membrane. Okay, so you can see here in arrows, yellow arrows, the direction of the protein product from the endoplasmic reticulum. They will be sent to the Golgi complex, which will then be packing them, ready for their travel to the duct. Okay, so not only will they be packaged, the protein hormones, but also modified as well. Okay, so they will then be sent into vesicles. Okay, they will be put into vesicles, and the vesicles are going to be traveling towards this polar portion of the cell where the duct is. Now notice there is a direction, there's trafficking, and the reason behind that, or the mechanism behind that, is very important for us to understand. Okay, but that will be a subject for later videos. And then once the secretory granules are here, and once the hormones are needed, or periodically the secretory granules are going to be merging with the plasma membrane of the cell in this portion near the duct, and the hormones will be released into the duct. And then here's an illustration of a cell that's also very polar in nature. We can see that this is one tip of the cell. You can see the nucleus in this weird position of the cell near one of the tips. Okay, and you can see the mitochondria as well. We have the RER, okay, for manufacture. And then from the RER, whatever is manufactured, perhaps hormones also, are going to be sent to the Golgi complex. And the Golgi complex are going to package those and put those in granules. And here we can see the granules. This is a mucigen granule. So this could be from a mucus cell lining perhaps your intestine or your stomach. So perhaps for the stomach, to protect the stomach lining from being eroded by the acid. So there are also mucus secreting cells that keep the surface coated with mucus. So the mucus here were made from the ER packaged in the Golgi apparatus and then sent to the outside through mucigen granules. So, let's now look at the mechanism of how the protein is made. Take note that proteins are polypeptides. Okay, so they are linear in their core structure. So here we can see a linear polypeptide. So this is the primary structure. And we know that in genetics, okay, in your lesson, that polypeptides are made through the process of translation at the ribosomes. So you have here your mRNA and your tRNAs involved. And through translation, this polypeptide is going to increase in length until it's done being made. Now here at the bottom, we can see a portion of the membrane of the endoplasmic reticulum, particularly the rough endoplasmic reticulum. And then there are receptors here, particularly your signal recognition particle receptor. And here we have a channel known as a translocon, which also has a gate, the plug. Okay, so when the polypeptide here, also known as a nascent polypeptide because it's just forming, okay, it's newly formed, is almost done forming, a signal recognition particle is going to attach to it. Okay, because of this signal recognition particle sequence, okay, that will sort of attract or complementary to the signal recognition particle. And that's very important because it's the signal recognition particle that will be recognized by the SRP receptor. And once that's done, the ribosome with the nascent polypeptide is now held in place right next to this translocon, okay, so that the nascent polypeptide can enter into the lumen of the endoplasmic reticulum. So the lumen is the space inside this membranous organelle, okay, the lumen of the endoplasmic reticulum. So the SRP leaves, okay, the signal recognition particle has done its job. The ribosome is depositing this polypeptide, newly formed ones, okay? And inside, there's also a chaperone protein waiting. And right there and then, the polypeptide is folded into its secondary, tertiary, and perhaps even quaternary structure, okay, as it enters the translocon in the rough ER particularly. And so this is how the protein synthesis is done and then in the ER. Okay. But take note, there are also proteins or ribosomes that are making proteins that do not need to dock on the ER, okay, or on the ER. There are also those ribosomes, but a majority need to dock on the ER, just like what we're showing here. So here's a picture of the translocon, and there's the plug. And then here you can see the outline of the ribosome. Okay, we have the large subunit facing down and the small subunit facing up. Okay, and here's your nascent polypeptide being pushed into the lumen of the ER as it's done, as it's finishing up in the translation process. So you see, it's very efficient. The ribosome doesn't wait for the translation to finish before transferring the resulting polypeptide into the RAR. The ribosome already goes to the RAR right away so that there's no problem depositing the nascent polypeptide. Okay, so how then are integral membrane proteins, no? If the nascent polypeptide is an integral membrane protein, meaning to say it goes through the lipid bilayer of the cell membrane, how then are they deposited? So in this case, the nascent polypeptide doesn't go all the way into the lumen, but instead there are certain charges thanks to the composition of amino acids, okay, in the nascent polypeptide that causes it to orient at this particular segment, okay, to align to this thickness of the membrane, the bilayer. And then this translocon is actually capable of opening sideways, sort of like a hinge. Okay? So the translocon opens sideways, and the nascent polypeptide, instead of going through, will just float through laterally. Okay? And then here you have your transmembrane or integral membrane protein. Or it's also possible that the orientation is of the reverse thanks to the reverse charges here in the amino acids of the nascent polypeptide. And so you have here another example of an integral membrane protein. The only difference is here on the left, the N-terminus is at the cytoplasmic side, and here the N-terminus is on the outside, if this is going to be the cell membrane. But in this case, this one is the ER lumen side, and this one is the cytoplasmic side of the cell. So, how do we maintain membrane asymmetry? Okay, so here's a simpler view of that transmembrane protein. So here's the membrane of the ER, here's the lumen, here's the portion where here's one side. Okay, we will discuss the difference between the sides later. Here's another side, another end. And see here how the vesicle is forming. This end with these are carbohydrates, is inside the vesicle. Okay, so as this moves into the Golgi apparatus, okay, the carbohydrates stay within the lumen of the Golgi complex. And the Golgi complex sends this to the cell membrane itself. And look where the ones that are in the lumen are found now. They are now pointed towards the exterior of the cell, towards the extracellular matrix, perhaps, of the cell. Okay? So, see in the endomembrane system, the portion that's in the lumen, or inside the organelle, becomes the portion that's facing into the exterior of the cell later on in the plasma membrane. So take note of that. That's important. Okay, so you can see here the cisternal. Cisternal is also the term for this lumen. Okay, luminal or cisternal are the same. Okay, so here we have a graph of a type of phospholipid or lipids in the ER. Okay, we can see here there are more PC or phosphatidylcholine than sphingomyelin in the ER. And then there's the least number of lipid would be the phosphatidylserine. But look at this now in the Golgi complex. Okay, the amount of sphingomyelin increases a bit more, the amount of phosphatidylcholine decreases, and this one also increases a bit. Now look at this in the cell membrane or the plasma membrane. There are now more sphingomyelin than phosphatidylcholine. So how is that possible when in fact in the ER there are more phosphatidylcholines, but in the plasma membrane there are more sphingomyelins? Okay, so look at the trend here. So the answer is there will be modifications in the lipids. Okay, the first one will be the modification through enzymatic activity. So here we have a type of lipid with a yellow head converted by an enzymatic activity into a lipid with a red head. Okay, so see the conversion. So in the plasma membrane, there could be more enzymes that would do this. Thus, there will be more red-headed phospholipids in the plasma membrane than, say, in this membrane layer. Second would be the selective selection of lipids in the formation of vesicles. So notice here, in this particular vesicle that's forming, there are more red-headed, okay, membrane lipids than yellow-headed. And look at the result. In the remaining lipids, there's at least more or less 50-50 yellow and red headed phospholipids. But here in this vesicle that is formed, there are more red-headed phospholipids. So that's one way of changing the composition of the lipids from ER to plasma membrane. Or there simply could be an enzyme that causes the flipping of this phospholipid. So from this side, it now has gone into the opposite side, and you know the name of this enzyme is called flippase, okay, because this movement is known as a flip-flop movement. So these are the ways by which membrane asymmetry can be accomplished in the different membranes of the cell.